Hello everybody, this is Jason from STEM Nerds. Thanks for being here. In today's video, we're going to talk about what are bacteria. They're all around us. They inhabit just about every single part of our planet. Let's go take a closer look. So first thing, bacteria and the archaea are part of a group known as prokaryotes. They lack a nucleus, they lack membrane-bound organelles, and they tend to be very small. In fact, they're smaller than the cells that we find in the human body, which are called eukaryotic cells, because our cells have a nucleus. They have membrane-bound organelles, and they tend to be larger. So bacteria are small. You're going to need a microscope in order to see them. As I've already mentioned, bacteria can be found everywhere, on surfaces, on our body, in all different parts of the environment. They can be different shapes. Right here, you're looking at a bacterial shape called bacillus or bacilli, and these are kind of oval-shaped bacteria, and we're going to talk about the structures here and all the things that happen inside of them here in a moment. Other bacteria are circular in shape, and that's coccus, singular, or cocci, plural. And again, these microbes can be found in all different places. And there are other shapes as well. The vibrio, kind of like a curved U, spirillum, kind of like a wavy little shape, and the spirochetes kind of look corkscrew-like. But the two most popular ones, or the two most important ones, are usually going to be the bacilli and the cocci. One of the things that make bacteria so important is they can be part of our normal microbiota or normal flora, and they can actually keep us healthy. These are microbes that line the internal and external surfaces of our body, and they can do things like outcompete pathogens for nutrients and take up binding places that keep us safe. They can make nutrients for us that we can't make on our own, and they can even make antimicrobial chemicals that destroy pathogens. However, we do have to be careful because there are some bacteria that are indeed pathogenic. We're talking about things like E. coli and Shigella and MRSA and these types of streptococci that can cause strep throat and wound infections. And so we have to take precautions, especially with the environmental microbes that wind up on the surfaces of our body. And that's why we do things like washing hands to wash away some of those potential pathogens. And of course, we make sure that we keep our surfaces relatively clean because microbes can hang out on those surfaces for a limited period of time. And if we touch surfaces that have bacteria on them and we wind up putting our fingers in our mouth or in our nose or in our ears, sometimes we have these what are known as portals of entry where the microbes can actually gain entry into the body. And microbes can be found in some very weird places. We're going to talk about it a little bit later in the video. They can be found, especially the archaebacteria, in very extreme environments, extremely acidic or alkaline, very, very, very hot environments, and even places where there is no oxygen. One of the main ways that we categorize bacteria is based on something called their gram stain. They can be either gram-negative or gram-positive, and it's based on their cell wall structure. And this can be really important for how we treat microbes, especially when they cause an infection. And right here, you're looking at a drawing of a gram stain. We've got these purple organisms. They're gram-positive, and the pink ones are gram-negative. And again, they can be different shapes. They can be the bacilli or the cocci. And so the gram stain results can tell us a lot about the microbe. Here we're seeing gram stain results for some of those bacilli. So structurally, what is the difference between a gram-positive and a gram-negative bacteria, you ask? Well, you can see both of the gram-positive and gram-negative, they have a membrane towards the bottom, that cytoplasmic membrane. But the gram-positives have this big, thick layer of what is known as peptidoglycan. And this is a mix of sugars and protein, and it basically is part of their cell wall. It comprises their cell wall. And so what we should know about gram-positive is they have a membrane, but exterior to that, they have a big, thick layer of peptidoglycan. That is their cell wall. Now, if we go over to the gram-negative on the left-hand side, they still have that cytoplasmic membrane, but then their peptidoglycan layer tends to be much thinner, usually just a single layer, maybe just a couple layers thick. And then exterior to that, there is an outer membrane. Now, what's interesting is both the gram-positive and gram-negative cells look different structurally, and that's the reason why we get the difference in the staining process, why gram-positives look purple and gram-negatives look pink under a microscope after you get done staining them with the gram staining process. But they also sometimes have to be treated differently when they cause an infection. The interesting thing is that bacteria, being prokaryotic, they have a relatively simple overall structure. And so we can see here, starting from the outside, we have the cell wall, we have a cell membrane. Some microbes, not all, will have an exterior capsule covering in addition to their cell wall. But when we travel inside, you can see there's not a lot going on there. They have DNA in that nucleoid, and even though that DNA looks kind of complex right now, it is usually just one large circular chromosome that's kind of folded up on itself, which is very different than what we find in eukaryotic cells. The other piece of genetic information that some or many microbes contain are things called plasmids. These are extra chromosomal, so outside the host chromosome, they're extra chromosomal pieces of DNA. And these plasmids can house genes that give microbes special abilities that are not necessary under normal conditions. One of the most important of which is antibiotic resistance. And these plasmids can be transferred from one microbe to another. You're going to see that they also may have mechanisms for motility or moving around. And so things like flagella or pili, and pili are really good at also, in addition to movement, attaching to surfaces and even helping exchange those plasmids through a process called conjugation. You're going to see that bacteria have ribosomes, and those ribosomes are useful for making proteins. But overall, they may have some vesicles and some vacuoles, and they might have a sort of a simple cytoskeleton. Another thing that they may have is something called a spore or an endospore, the ability to form these endospores when conditions are less than optimal. And at that point, the cell can go into almost a dormant state where there's not a whole lot of metabolism happening, but the spore protects the microbe from UV, from extreme temperatures, from desiccation, which is another way of saying drying out. And the spore can eventually break down when the microbe finds itself back in optimal conditions. Now, microbes replicate via a process called binary fission. Basically, again, we're dealing with a cell that is more simple than a eukaryotic cell that we would find in human beings. And for us, we talk about the process of mitosis. Binary fission, not quite as many steps. And if we look at it from a simple perspective, you've got a cell that copies its genetic material and then splits in two. Now, binary fission can happen very, very quickly for bacteria in ideal conditions. Cells, E. coli as an example, they can replicate every 15 to 20 minutes in ideal conditions. In nature, usually the replication is a lot slower, and there are many microbes that can take hours, days, even weeks to replicate. Now, I mentioned that microbes have the ability to exchange genetic information, and these are the three main mechanisms: transformation, transduction, and conjugation. Transformation is interesting in that usually what can happen is when bacterial cells in an environment die, they lyse. And when they do that, they kind of spill out all of their internal stuff into the environment, and cells that have the ability, in other words, cells that are competent, can take in some of that genetic information from the cell that is lysed. And that process is called transformation, the ability of a cell to take in genetic information from another cell. And if that genetic information provides some type of an advantage, that genetic information may be maintained within that cell. And every time that cell grows and divides, that new genetic information will remain. I'm going to skip to the bottom. The other one is transduction. Here, this is a little bit different. It's kind of interesting, but yes, even bacteria can be infected with viruses, and that's that little space alien thing that you see down there. That is called a bacteriophage. And what can happen is a bacterial cell gets infected by a virus, and the viruses don't know any better. They just start incorporating genetic material into that protein capsid, that space alien looking thing. And sometimes they accidentally incorporate the host bacterial DNA into their capsid instead of viral DNA. And then when they try to infect a new cell, they accidentally inject the bacterial DNA into the new cell instead of viral DNA. And so the recipient is like, oh, okay, well, this isn't the end of the world because I've just got new DNA from another bacterial cell. It's not an infection. And again, if that genetic information provides an advantage, it may be maintained within that cell and then the subsequent cells that divide from it. Perhaps the most interesting and potentially most dangerous way that bacteria exchange genetic information is via this process called conjugation. Now, here, this involves pili, and the pili make a connection from one cell to another. And the cell that initiates the contact, we label that F plus for fertility factor. It has the ability, because of a plasmid, to make a connection with another cell and transfer that plasmid from itself to a recipient. That recipient cell before the contact was labeled F minus because it didn't have that fertility factor. But after the process, what happens is the initial cell, the initiating cell, will basically transfer over one strand of its plasmid to the recipient, and they'll both make the complementary strand of the plasmid. And now we've got both cells that have the plasmid. They're both F plus. They're both capable of transferring that plasmid to another cell. Why does this matter? Because this is a primary way in which antibiotic resistance can form. Antibiotic resistant cells that carry the antibiotic resistant genes on those plasmids have the ability to transfer those plasmids to another cell, thereby making them antibiotic resistant as well. What makes matters even worse is sometimes the cells that are initiating that contact, they don't actually infect human beings, but the cells that they're contacting might. And so this is something that we are keeping a close eye on in the scientific community and in the health community, because when you have the transfer of plasmids that allow for antibiotic resistance, that can create antibiotic resistant infections that are very difficult to treat. Now, interestingly, I did mention that microbes can live in some extreme environments, and I want to take a few minutes and just kind of show you how we classify microbes based on where they're living. And so we can classify microbes based on temperature. Microbes that like really, really cold temperatures are known as psychrophiles or psychrotrophs. These are microbes that are living in the Arctic. These are microbes that might be spoiling your food in your refrigerator. Microbes that enjoy human body temperatures are called mesophiles. And microbes that like really warm temperatures, like you see here, this is the base of a thermal vent deep in the ocean where there's volcanic activity. And this water right here can actually be above the boiling point. Microbes actually live there, and they're called thermophiles. Thermo, heat. Phile means to love, so either thermophiles or extreme thermophiles. And so we can classify microbes based on the temperatures in which they inhabit. But we can also classify microbes based on the pH that they enjoy. And so we've got these microbes called acidophiles, and this would be an example of an acidic environment. But there are also acidic environments even inside the human body, such as the stomach or the lower female GU. And so acidophiles love acidic environments. They may grow anywhere from a pH of 1 to up to 5, where most other living things are going to struggle. Neutrophiles are the microbes that we tend to be most concerned with as human beings, because most of our body is around a neutral pH. And so the neutrophiles, usually somewhere around between 6 to 8 is where they're going to thrive. Our body is buffered usually right around a pH of 7.2, except for the stomach and the lower female GU. Then we have the alkalophiles, and they like a higher pH. You might find those in the soda lakes where our pH is above 8, usually going up to 10, maybe as high as 12. You can also classify microbes based on whether or not they require high salt concentrations. And a lot of microbes, especially the ones that are going to interact with human beings, are what are known as non-halophiles. Halo means salt. And so these are microbes that really don't want a lot of salt. They're microbes that are going to associate themselves with the human body, with the exception of the skin, and microbes that you might find in freshwater environments. The halotolerant microbes, these are microbes that can tolerate high concentrations of salt, and often these microbes are found on the surfaces of our body that can have a salt concentration as high as 9%. Halophiles, these are microbes that need additional salt, and so these are microbes that you might find in the ocean with a consistent salinity of around 3.5% sodium chloride. And then you've got what we're seeing here, the extreme halophiles, and these are microbes that need salt concentrations as high as 20 to 30%. There aren't that many places in the world that have salt concentrations that high, and most living things cannot live in those environments, but there are microbes, especially the archaebacteria, that can survive and thrive in those high salt environments. The last environmental parameter I want to mention today, and that is microbes can survive in different oxygen concentrations. And so there are microbes on the far left-hand side that like oxygen, just like we do. And we call those microbes obligate aerobes because they're obligated to be in an environment where there is oxygen at normal atmospheric concentrations, which is right around 21%. So they would be very similar to human beings, and they would be found in the same places that we're found. Now, on the opposite side of that, right in the middle of the screen, we've got the obligate anaerobe. And this is a little weird because we're not used to seeing something like this, but these are microbes that do not like to be around oxygen, and in fact, they can die in the presence of oxygen. Now, the tubes that you're looking at here, these are actually a thing called nutrient agar. And as this nutrient agar media is solidified, there's more oxygen near the surface and less as you go towards the bottom. So there's an oxygen gradient. Now, the obligate anaerobes, they have to be in low or no oxygen environments. And the reason for that is they lack enzymes that actually detoxify oxygen. Sounds weird that oxygen could be toxic, but it can be for certain microbes that lack certain enzymes. And so the obligate aerobes have enzymes that allow them to break down oxygen radicals. The obligate anaerobes do not. In the middle, we've got the microaerophile. Micro means small, aero, oxygen, phile means to love. So they love small amounts of oxygen. And so they have these enzymes that will help them detoxify oxygen radicals, but they don't have them in the same concentrations as the obligate aerobes do. Therefore, they need some oxygen, but not as much as an obligate aerobe. And so you might find them in areas maybe a little bit lower in the soil where there's not as much oxygen there. Typical oxygen concentrations might be in the high single-digit percentages, so maybe 8 to 9% instead of the 21% that we find with the obligate aerobe. Now, the two on the end, at first glance, look very similar, the facultative anaerobe and the aerotolerant anaerobe. These are interesting organisms because the facultative anaerobe prefers oxygen. But if oxygen is missing, it switches to fermentation. And that's why you see here in this tube, it grows better when oxygen is available. But as you see, as you go down the tube, there's still growth there because the microbes are switching to fermentation. They don't produce as much energy, but at the same time, they're able to remain alive. The aerotolerant anaerobe on the far right-hand side, you're going to see that they also grow throughout the tube. They can only ferment, and so they lack the enzymes, they lack the structures necessary to grow in the presence of oxygen and actually use it. And so they, unlike the obligate anaerobes, they can grow in the presence of oxygen, but they're not killed by it. Unlike the obligate aerobes, they can grow in the presence of oxygen, but they cannot utilize it. And so they're going to be found pretty much throughout that entire tube fermenting. They're strict fermenters. Finally, we can classify microbes based on how they eat. And so the two primary ways that we can do this is we can describe microbes as chemotrophs. In other words, they can consume a chemical source of energy. And in other words, they're very similar to us. We are chemotrophs, and if we take it a step further, if we know that an organism is using chemicals as a source of energy, we say, okay, well, what kind of energy? What type of chemical? Is it organic, carbs, lipids, and proteins? We use organic compounds as a chemical source of energy, and therefore we are called chemoorganotrophs. Troph means to eat. And so we are chemoorganotrophs. Well, there are lots of microbes that do the same thing. They consume organic compounds in the form of carbs, lipids, proteins, and so they are also chemoorganotrophs. But interestingly enough, there are other microbes that still consume chemical sources of energy, but the chemicals are inorganic. They're not carbon-based. I've got a picture here of a fish tank filter, and these microbes are really important when it comes to making aquatic environments safe for fish. And so our chemotrophs that don't use organic compounds, in other words, they're using inorganic compounds, are called chemolithotrophs. Really good examples of what they can eat, and the reason why I have this picture here, living things in aquatic environments produce ammonia. Well, there are microbes that can eat ammonia. That's what they're using as a food source. So that is inorganic. Nitrite would be another example. Sulfur compounds would be another example. And so these are microbes that are using a chemical, but they are not carbs, lipids, or proteins. And so we talked about the chemotrophs, but there's another mechanism, and that is phototrophs, like these cyanobacteria here. They're very similar to plants. They're using visible light in order to make energy. Now, one last thing, in addition to describing the energy source of a microbe, we can also describe, well, how does the microbe build its structure? How does it make new cells? How does it use carbon for building structure? And there's two words that we can use, one of which is heterotroph, and these are microbes, just like us human beings, that have to consume our carbon to make new cells and to make us grow larger. Heterotrophs are organisms that need to consume it in the form of carbs, lipids, and proteins. But going back to our cyanobacteria, they are autotrophs, and they can fix CO2 like plants and like algae. And what an autotroph does when we say they're fixing CO2, what we're saying is they're taking the CO2 out of the atmosphere, and they are converting that CO2 to the carbs, lipids, and proteins that they need in order to build structure. All right, everybody, so that was a crash course on what bacteria are, how we classify them, where we find them. As always, thank you so much for being here. Have a great week.